All right, guys, so the question that we're looking at for this specific problem is trying to figure out what amino acid is going to be coded for by that specific DNA sequence of G, A, T. And so the first thing we kind of have to do is take a look at this codon chart that's off to the side here and notice that my DNA sequence has the letters G, A, T. Well, in this codon chart, I don't see any T's anywhere. So I know that this codon chart isn't going to be working with DNA. I do see a U over here, which means it's definitely going to be working with my RNA molecules instead. So the first thing that I have to do is actually transcribe my DNA into my mRNA molecule. Remember, this is our messenger molecule. This is the part that can leave the nucleus and can actually get taken to the ribosome in order to do protein synthesis. So my DNA is GAT, and my mRNA is going to be the complement of that. And the only difference between trying to make the other strand of DNA and my mRNA is going to be that I'm not allowed to have any T's anywhere. So anywhere I would have put a T, I'm going to put a U instead because uracil is that molecule we're allowed to use. So I know that in DNA, G usually pairs with C. That's our standard practice with our DNA to DNA. Well, C's are totally allowed in my mRNA molecule. Those are a molecule I can have. So I know that this first letter of my mRNA is going to be a C. The second letter of my DNA molecule is an A. Now, normally I would pair A with T, but my RNA molecules are not allowed to have thymine. They can't have T. So instead, I'm going to pair it with a U. And then the final letter of my DNA sequence is a T. And T usually pairs with A. I'm totally allowed to have an A in my RNA molecule. So we're going to go ahead and add an A there. Okay. So this is my RNA sequence, my CUA. And that's what I can actually go through and use for this codon chart that's over on the side. So now we need to figure out what our amino acid is, which I'm going to abbreviate with AA for amino and then acid. And now I have to actually look at this table and use this mRNA sequence to answer the actual question. So what I do is I start from the very center, just like it says down here, move from the center circle out. And so the first thing I'm going to do is out of this bigger center circle that's here where it says G, U, A, and C in really big letters, I'm going to find the C. And I can see that I've got the C right here, which means I'm only going to be working in this corner of this kind of pie chart that we've got going. So I've already eliminated, not going to be using this because that's for G, not going to be using this because that's U, and not going to be using this because that's A. So I'm only going to be using this bottom corner here. And so now that I know that C is here, now I have to look at these four options and figure out what the next letter is. And so I already coded for that. It's a U. So I go from C and then I go to the U that's right there. And then now I'm only going to be working in this quadrant. So I kind of already know the answer, but we're going to keep going anyway. So I've got C, then U, and then I've got my A, and that's going to code for the amino acid leucine, which we can actually just abbreviate with the first three letters. That makes it a little bit easier, so you don't have to write out the whole thing. But that's essentially how you can go forward and actually take a look at these types of things. You start from the very, very center, and you work your way out.